In this video we are going to show DNA replication. So this is a double-stranded DNA molecule. One strand is from 5' to 3' and the complementary strand is from the other direction, 3' to 5'. Now for the DNA replication to occur, these two strands must separate. So when these two strands start to separate by specific proteins called DNA helicases, they form a Y-shaped structure which is called DNA replication fork. So this is Y-shaped DNA replication fork. So DNA helicases started to unwind these two strands and you can see they are separated. Now DNA replication will start by specific proteins called DNA polymerases. So DNA polymerases synthesize DNA from 5' to 3' direction. So that means it will start from here. The 3' end is in this strand. So when it will start, 5' end will form complementary base pairing. So accordingly, I'm going to show you how exactly it looks. So the DNA polymerases started to synthesize DNA from 5' to 3' direction in the newly synthesized DNA strand, and this newly synthesized DNA strand is made based on the template strands nucleotide sequences. So this original strand, which has separated from the other strand, is called the template strand, and so based on this template strands nucleotide bases, so T paired with A, C paired with G, G paired with C, A with T, T with A, T with A, towards 3' direction, DNA synthesis has started. And this strand where DNA synthesis has started from 5' to 3' direction in a continuous fashion is called leading strand. For DNA synthesis to occur in the other strand is little tricky, because here the end is 5', and as the DNA synthesis occurs from 5' to 3' direction, it can't start actually from this. In the newly synthesized strand, it can't start from 3' end and go to this direction. Rather, it has to start from a 5' end and proceed towards the 3' end, and that's why this DNA synthesis in this sequence is discontinuous. So when the two original strands they separated, it can start from the place where it is already separate, but from the other strand, which is I called already the leading strand, as the unwinding goes on, the synthesis of DNA can continuously go on as the direction is same and it is towards 3' direction, 5' to 3' direction. So the DNA synthesis in leading strand is continuous. However, DNA synthesis in the other strand, which is called lagging strand, is discontinuous. So when it opened, the DNA synthesis started from 5' to 3' direction. But how about DNA synthesis in those places? So when they will unwind further, the DNA synthesis will start again and it will proceed in discontinuous fashion. So in the lagging strand, the DNA synthesis occurs in discontinuous fashion. So initially I showed only up to this area the DNA synthesis could occur in the newly synthesized DNA strand. But as they started to unwind further, the DNA synthesis could again occur from 5' to 3' direction. So 5' end here, and based on this template strand, C to G, C to G, A, T, A paired with T. So here you can see that 5' to 3' direction it started to proceed this way. And there is a gap, and this gap gets later annealed. So ensuring that the DNA synthesis is complete. So these pieces of DNA that get synthesized in short stretches of DNA, they are also called Okazaki fragments. So as I have shown here, in the leading strand the DNA synthesis is continuous. In lagging strand it occurs in discontinuous fashion. And this is the Y-shaped replication fork. Hope you understood. Hope you enjoyed the video. Thank you so much for your time.